Today, we're very excited to have Doctors Josephine Forbes, who's actually an MD PhD, and Dr. Sherman Leung, who's a PhD, joining us from University of Queensland to discuss All the Rage, a novel target for prevention of type 1 diabetes. Just a quick bio on both of them. Dr. Forbes is a translational researcher performing bench-to-bedside studies on novel therapies to prevent type 1 diabetes and a major complication, kidney disease. Currently, she's based at Mater Research, the University of Queensland in Australia, and she is the Australian Diabetes Society President-elect, the previous chair of the Diabetes Australia Research Program, and an editor board member for journals including Kidney International. She is also continually in the top 2% of scientists globally in Stanford University's annual listings with an H index of 61 and has published 200 research articles with over 15,000 citations and 3 patents. She's received numerous prizes for her research, including the Commonwealth Health Minister's Award for Excellence in Health and Medical Research in Australia, and we're very excited to have her back. Our first conversation with her was just at the start of the pandemic in 2020. Dr. Sherman Leung, a quick bio sketch on him, is the head of research operations at Wesley University Institute in Australia, which is the official research partner for Uniting Care, encompassing the 4 Uniting Care hospitals in Queensland, as well as Lifeline, Blue Care, and Family and Disability Services. He's a PhD trained scientist, conducting his research training under the leadership of Dr. Forbes at Mater Research, University of Queensland. His recent work on the preventative therapy for type 1 diabetes has been published in Diabetes, receiving the cover image, ooh, that's awesome, and an In This Issue feature, and shared across the Australian-wide 7 Network News. He has extensive experience in clinical trials, having worked at Microba Life Sciences, an ASX-listed biotech startup, and Nucleus Network, Australia's largest early-phase clinical trial site, having overseen the conduct of several high-profile COVID-19 projects, including that of Nuvaxoid and Novavax, that is now authorized for use by the TGA, EMEA, and FDA across many jurisdictions worldwide. Thank you both very much for joining us. I'm really excited to hear about the work you've been doing down under there in Australia.
Thanks, Monica. It's our very great pleasure to be presenting to you today. And what we're going to do is present some of our research program, which is around all the rage and why rage might be a novel target for the prevention of type 1 diabetes. This is certainly a slide that you'd all be familiar with, and we know that diabetes is a major global problem. It's a pandemic. It doesn't get as much attention as COVID, but in all seriousness, it needs to be starting soon. But what you probably don't appreciate as much is why we study type 1 diabetes. Now, we all know the patient burden, we all know the importance of this disease, but some other underappreciated facts are that actually, even though it only makes up about 10% of the cases, it's around about 40% of the economic burden of diabetes to most governments. So it actually also costs our healthcare budgets a lot of money. It's a very complex clinical management. It often has an early life onset. It is increasing globally in westernized nations, particularly in countries such as China, India, and Saudi Arabia. And certainly what comes with type 1 diabetes is a risk for early death from complications such as high and low blood sugar emergencies and often kidney and cardiovascular disease. This is a slide that you're probably extremely familiar with, but we all know that there are a number of stages that are happening in type 1 diabetes before the clinical diagnosis. And this has enabled us to think about intervening, I guess, in some of these stages and what the pathogenesis might be, and what are some of the unique targets that we might be able to look at to provide therapeutics to stop this progression to clinical diagnosis. So the receptor for advanced glycation end products is probably something you haven't heard too much about in type 1 diabetes per se, from an immunological perspective, but certainly in complications of diabetes, this has been a major player with respect to the pathogenesis of things like kidney and cardiovascular disease, because it's involved in the development of vascular complications, particularly atherosclerosis and kidney disease. The receptor for advanced glycation end products is a very promiscuous receptor. It has a number of very important physiological roles. It's involved in host pathogen defense, T cell priming, antigen presentation, in particular by dendritic cells and other major antigen presenting cell subtypes. And it has a critical role in macrophage recruitment. You can see here that it has a number of different ligands, and we think the conditions that it binds to these ligands under are very different. For example, HMGB1 and the S100 calgranulins are probably the initiators of things like your standard inflammatory and immune responses. So they're part of those DAB processes. And advanced glycation end products are something where we believe it needs to hit a particular threshold to bind to RAGE. Something else you probably don't know about RAGE is that it's actually located in the HLA region. So it lives in the MHC class 3 region, right in between MHC class 2 and 1, which we all know is a major susceptibility loci for the development or susceptibility to type 1 diabetes. So my slides aren't advancing as fast as my mouth is talking. And so one of the things we were interested in, because this RAGE does sit in this particular region, we were interested in seeing whether polymorphisms in this gene actually were associated with the development of type 1 diabetes. And indeed, we could show in a population where we had control for the HLA. So obviously, we were thinking that perhaps RAGE might be co-inherited with some of these things. So we wanted to understand and unpack that a little bit more. So what we did was we only looked at people who had the same HLA risk in their background. And we could see that two particular polymorphisms increased risk for type 1 diabetes development. And there was a third one, which actually lowered the risk for type 1 diabetes. And so this was particularly interested. These are quite a number of, so there was about 641 cases, but this is a nested case control study from a larger cohort, very generously provided to us by Mikael Neep and by the Henrik Group, who are both in Finland, which has the highest incidence of type 1 diabetes in the world currently. What's most interesting about these particular genotypes is, though, or these SNPs, is that they actually are functional SNPs, and they change the concentration of a protein called circular RAGE in the bloodstream. And so those that provide susceptibility to type 1 diabetes, such as this particular change, actually provides, or people have lower amounts of soluble RAGE in their blood when they are from a population who is at risk of developing type 1 diabetes. So these particular samples are taken from young children before, or those who we know develop type 1 diabetes later in life. Something else we know is that those that actually encode for protection seem to either remain unchanged or actually have higher levels of soluble RAGE in their circulation. So we know not only is this change, but there is actually a functional outcome. And a very talented postdoc who is also a pediatric endocrinologist in Mikael Neep's group did a project that we were also involved with, looking at soluble RAGE in identically matched patients. And so this was a really hard study to recruit for, because all these people are exactly the same age, they have exactly the same duration, they have the before seroconversion samples. And we could show, in fact, that during the seroconversion stage and then over the next 3 years, soluble RAGE actually decreased. And so this was where we were looking approximately in that study I showed you previously at the concentrations. And you can see quite clearly that 3 years after seroconversion, something's going on. But certainly at seroconversion, it's on its way down. We also know that the plasma soluble RAGE in another nested case control, which was taken from 16,000 individuals, but the nested case control was 1,000 individuals. And so it showed that there was an association between soluble RAGE and the severity at presentation of type 1 diabetes. So it also told us that during that period where soluble RAGE was changing, there was also this impact on the severity in which type 1 diabetes was presenting as. What was also very nice, and this is some of Sherman's work, and this is unpublished data, but this is from their NPOD, the Network for Pancreatic Organ Donors. We didn't know whether RAGE was actually also expressed in other tissues, such as the pancreas. And so we actually had a look at this in patients with type 1 diabetes, and we could clearly show these are controls in the blue. These people here have autoantibodies, and these people here have type 1 diabetes. And so we could clearly see that there were changes perhaps in the ligands for the receptor for advanced glycation end products, and a significant increase from autoantibody positivity in the amount of RAGE in the islet. And we'll come back to that a little bit later and talk some more about that and why that might be important. But another experiment that we did was actually a failed kidney experiment that I did, where I wanted to induce kidney disease by injecting a RAGE ligand. And so we actually did it in two ways. We injected AGEs at a level where we thought we could reproduce the level seen in diabetes. And we also fed AGEs because they can also come to you from the diet. And so that's another particularly important point. We're not going to dwell too much on that today, but you can actually intake advanced glycation end products in quite significant amounts from a Western diet. And what we also did was intervene with an anti-AGE or an age-lowering therapy in this context. So what we could see is that, in fact, the AGEs, the RAGE ligands, did, in fact, go to areas that we thought were important for the development of type 1 diabetes. So the spleen and the pancreas, as an example here. What we also showed was that this was not the case in our mice with an AGE knockout, a RAGE knockout, my apologies. What was even more important, though, is that we actually saw three major things that we didn't expect. So again, remembering that this was designed as a kidney experiment, but we were looking at glucose control at the time as well. And we showed quite clearly that, in fact, there was an impairment particularly in first phase insulin secretion. And we all know that's an important step in the development of type 1 diabetes. There's this autoimmunity, there's this loss of first phase insulin secretion, and those two factors together are very strong predictors of who is going to develop type 1 diabetes. And so we can see both the high age diet and a high dextrose diet, which is a high sugar polymer diet, which also increases your levels of AGEs in the circulation almost to an equal extent. And we could see that there was a very big change in this insulin secretion. And we could also see that that was reversed with allogebrin chloride, which is an AGE lowering therapy. But what was even more surprising was that we could see that there was this infiltration. Now, in this instance, this is macrophages because this is what we were looking at the time. But you can see that it looks familiar to us in type 1 diabetes, that there's this peripheral infiltration of particular cell type, and that it's also seen inside the islet, and this is not there in the other groups.
Can you just comment on what was that drug that you used that's used?
Yeah. So allogebrin chloride is a very sad story. It got to late phase 2 clinical trials for diabetic kidney disease. So it was being tested in patients. It was also being tested for heart failure. And unfortunately, the financial crisis, I think the company was called Synvista, and the global financial crisis took away, or they fell to the global financial crisis. And because of that, what happens is the patent is tied to the bankruptcy of the company. And so the patent is essentially lost until it runs out of steam. So that's a very sad story of a very efficacious compound which got lost. And that's an important thing to remember. When we're thinking about drug development, there are all sorts of things that have to be considered along the way. And Synvista was very actively pursuing both of these indications, and the data we had were very promising from a diabetic kidney perspective. So yeah, it's a shame.
Is that company based in Australia, or was it based?
No. So it's a US company. It was based in New York. Yeah, so it started as Altion Corporation. So they had a couple of other drugs which were not so great and not so specific, but this one was much better.
And I wonder if you happen to know off the top of your head, when does that patent,
in the next couple of years, I believe, because we did have a conversation about what happens when that comes off patent? Do we have the ability to then chase that again? And certainly I'll show you some data. I think it's on the next slide, actually. We might go to the next one because I'll show you actually that it reduces the incidence of type 1 diabetes. So this is it here in the thing here. This is a low AGE diet, but the low AGE diet here was only given for a short period of time. So just letting you know that we have given low AGE diets for longer periods of time and not taken them away, and they actually also do improve the incidence of type 1 diabetes. And certainly this HMGB1 ligand here, this is other data from other people, but I think there's another really good paper in Diabetologia by another team showing that if you intervene and reduce this particular RAGE ligand, you can also interrupt the course of type 1 diabetes. I'll just sneak back to this one for 1 second. Our data up the top is not as exciting as this data, which is a study that was done in the UK. So it was done in school children. They were very large numbers. I think, Sherman, correct me if I'm wrong, but it was 3,000 or 4,000, and there were also twin pairs in this as well. And what they could show that on top of autoantibodies as obvious and known risk factors for the development of type 1 diabetes, the advanced glycation end product CML was an additive independent predictor for progression to type 1 diabetes in these children in the general population. So that was a very nice study that was done some time ago by this team here. So needless to say that it was really important to us to think about RAGE antagonists. Now, at this time, we already knew that allogeneic chloride was not going to be available to us to continue to develop for type 1 diabetes, even though it was our compound of choice at the time. So we needed to think about whether or not we could target RAGE. And so Sherman, I think I've got a couple more to talk about, but look, we know from our studies in kidney and cardiovascular disease that RAGE antagonists have a number of effects on the body. We anticipate that they will have some preservation of beta cell function, because this has been shown in models of type 2 diabetes where RAGE has been knocked out or in fact altered in terms of its expression. And we also know that it has a number of modulating effects on particular cells involved in the development of type 1 diabetes. And it's also been shown to have a role in glucose tolerance. So there's a particular process in the muscle in particular, which involves RAGE in terms of the take-up of glucose in terms of that pathway in the muscle, and it's turning into glycogen. So this is a study just showing when we give soluble RAGE, where does it go in the mice? So it certainly goes to lots of places, and some of that data is under review, so I'm not going to show it today, but certainly this is one part of that data. And we can show that when we inject soluble RAGE, this is just a single shot injection that we're showing here, that it in fact does go to the pancreatic islets. So we actually can see it there. You can see it here. This is the red, insulin is the green, and obviously this is the immune cell infiltrate here. So we do know that it traffics to areas that are important for the development of type 1 diabetes. So it's not just about what's in the circulation, it's also about what's going to the tissues which are important for the development of type 1 diabetes. What we had also previously published in that space was thinking about acute therapy for diabetes and whether or not we could give soluble RAGE. Being a biologic, we didn't want to give it for a long period of time. Really important because people make autoantibodies and other things happen which are undesirable side effects for particularly young people. We don't want to give something where the side effect profile is not acceptable. You know, these people, we want them to live a long, healthy life, productive life. And so what we do know in NOD mice, which is a model we use sometimes to understand the development of type 1 diabetes and what might be useful, is that we can see that, in fact, in NOD mice, they don't actually have a lot of soluble RAGE. And if you look at their SCID counterparts at the same age, they actually have quite a lot of soluble RAGE. Now, this may be a nuance of the two particular subtypes, but that certainly piqued our interest as well, and certainly went along with our hypothesis. So, Sherman, this is for you. So I'm going to hand over now to Sherman, who was the first author on this paper, to present some of his absolutely beautiful data.
Thank you, Jo. So over the next few slides, I will share a bit of data that has contributed towards the SRAGE treatment paper that's now published in the Diabetes Journal. As you can see on screen, there's two diabetes incidence curves, and essentially in these experiments, what was conducted was the NOD mice, which spontaneously developed type 1 diabetes or type 1 diabetes-like disease. They were either given a vehicle treatment, a control arm, or they were given one of two types of SRAGE regimens. And this was either SRAGE given twice daily at a dose of 25 micrograms, or it was SRAGE given once daily at a much higher dose, 100 micrograms. And most importantly, as Jo highlighted before, we were looking at giving this therapeutic in a very brief or defined therapeutic window from days 50 to 64 of life, as you can see on screen in the top left-hand corner. And following that treatment, we didn't treat any further and observed for the onset or development of autoimmune disease. And what we can see in these diabetes curves by those two graphs in the center of the screen is that certainly when mice were administered SRAGE, as seen by the red lines, there were significantly less mice that developed T1D. And this was reflected across two independent research sites, one in Australia, which was managed by ourselves at Marta Research, as well as one other site in the US, which was managed by a pharmaceutical partner. And probably most interestingly, we almost see a dose-dependent effect where if mice were given S-RAGE at the higher dose seen by the brown bar, we do almost seem to have even less T1D onset, which is really reassuring to see. One thing we were really interested in looking at as well, apart from the onset of T1D itself, was the development of autoimmunity in the pancreatic islets. And as you can see on screen in the very top, there's a series of pancreatic islet images, and going from left to right, these are islets at increasing level of islet infiltration by the immune cells, and the dark purple cells certainly reflect the immune infiltrate within the islets. What we did see when mice were given S-RAGE through the graphs at the very bottom is that certainly the infiltration of islets significantly decreased if the mice were given S-RAGE therapy, which is consistent with the decreases in T1D onset that we saw in the previous slide. And that was very reassuring. We saw this as a measure of proportion seen in the graph on the left, as well as the overall insulatus index seen in the graph in the center. And at the end of the study, when we actually looked at the number of islets, the total number of islets that we could count and quantify, mice that were given S-RAGE actually had far more islets per tissue area, which was also very reassuring and consistent with the ability for S-RAGE to modulate the onset of T1D in mice. Shown on screen here is a very seminal study, which I think most people are very aware of, which was led by Professor Kevin Herold in the US, as well as the Type 1 Diabetes TrialNet group, and certainly very pivotal data that showed that in relatives at risk of T1D, if they were administered teplizumab, an anti-CD3 antibody, that this disease-modifying agent was certainly able to decrease the onset of type 1 diabetes as compared with the placebo arm. So very encouraging evidence, and certainly demonstration that therapeutics which target, in particular in this case, T cells, are able to modulate the onset of disease pre-diabetes.
Yes, I would also comment that that's been, even validated even more yesterday with Sanofi's purchase of Prevention Bio, and one of their for $2.9 billion, and one of their assets was the TZ Shield, which is the teplizumab asset. So I think, right, so this is a really great space for you guys to be in, because there's an opportunity, right, for endotypes and for multiple, treatment or, multiple druggable targets. So it's very interesting to hear what's next with you.
Absolutely, fully agree. Very exciting, Monica. And I guess what this slide shows here on screen at the moment is a bit of a schematic as to the a few of the type of major immune cell subtypes which are involved in T1D onset. And some of these obviously were able to be modulated by teplizumab. And some of the ones that I'll highlight include the regulatory T cells, the Tregs, including the iTregs, which interface with some of the other immune cell subtypes, such as the effector T cells, the T effectors, as well as the antigen presenting cells. In addition to that, obviously, they contribute towards the production of certain anti-inflammatory milieux, such as your cytokines. So if we go on to the next slide, how does this interface with the work that Josephine and myself worked on in this project? Well, certainly many of these immune cells express the RAGE receptor. And shown on the very left is a diagram as to how the RAGE receptor actually binds various ligands. It includes ligands like your advanced glycation end products, AGEs, as well as HMGB1, your calgranulins, and a few other well-known, typically acknowledged as pro-inflammatory ligands such as IAPP and LPS, and certainly the use of soluble RAGE or S-RAGE appears to scavenge these ligands, as seen by the soluble RAGE schematic on the top left. And some of the literature which has come out to date, if you can click next, certainly demonstrates that many of these immune cell subtypes do express RAGE, and these include, for example, the dendritic cells, which depend on RAGE for in vivo homing to the lymph nodes following antigen recognition. The maturation of the DCs also appears to be influenced by one of the RAGE ligands, AGEs. The T cell activation process also appears to be modulated by RAGE, and this is shown, I believe, in human T cells. This is the Journal Immunology paper, the second from the bottom. And also, most recently, Professor Herold and his group in the US also found, these are in type 1 diabetes samples, that the RAGE receptor, in particular gene expression profiles for survival and inflammation, were modulated by that pathway as well. So very exciting data that shows that RAGE may have an important role to play in T1D development. So after we looked at the onset of T1D itself and the infiltration of the pancreatic islets in our mouse models of type 1 diabetes, we wanted to look at the immune cell compartments to see if we were able to modulate the regulatory T cells as a proportion relative to the T effector cells, because certainly many disease-modifying agents were appearing to look at this arm or this angle, including, for example, tibulizumab. And what we see on screen is a graph of characterizing two main lymphoid compartments, the pancreatic lymph nodes, as well as the spleen. And this is immediately after the therapy of SRAGE was given at day 64 of life, shown with that bright green arrow. And reassuringly, what we do see here is that in the local draining lymph nodes, the PLN, certainly the ratio of Tregs to the effector T cells increased. And as a crude measure, certainly seemed to suggest that there is an increase in an anti-inflammatory environment within that local environment. And while we didn't see this in the spleen, we certainly did see it in the pancreatic lymph nodes, which provided some data that perhaps immunologically, SRAGE was modulating the development of T1D.
Yeah, that's
we move on to
fantastic.
If we move on to the next slide, the next thing we wanted to look onto was how Tregs or the immune cell infiltrate look like in the islets themselves. Certainly, we looked at the infiltration as I guess, a marker overall using H&E staining, but we wanted to see specifically if the regulatory T cell compartment within the islets change themselves, which is exactly where the damage tends to occur in type 1 diabetes. And to do this, we performed some immunofluorescence staining using many immune cell markers, very standard ones. And after quantification of the amount of Tregs within the islets following SRH therapy, what we did see was that certainly immediately after the therapy was given at day 64 of life, as well as at the very end of the study at day 225 of life, the percentage of Tregs did significantly increase within the pancreatic islet tissue itself, which is reassuring. This associated with an increase in the insulin area. Again, something that one would probably positively associate with preventing the onset of type 1 diabetes. And shown in the very bottom left-hand corner, if we correlate these two measures, the percentage of Tregs against insulin area, we did see a positive association, which is something that we would have liked to see, which also tends to suggest that these two increases in Tregs and insulin appear to be associated in some manner.
Sherman, quick question from the chat. How do you define the CD8 positive Tregs?
Yeah, great question. So our definition of Tregs has been using standard markers, so CD3, CD4, FOXP3. I believe in this project, we didn't look specifically at the CD8 Tregs, but certainly that was one of the areas for future investigation that we were potentially interested in. So great question.
Yeah, I could chime in there too. We certainly concentrated on the CD4 T regulatory cells for this paper, but we do know that the CD8 regulatory cell compartment also looks very interesting, and we're actively trying to design a more strategic panel to actually look at that a little bit better, because the panel we were looking at was very much towards the CD4 compartment.
Great, thanks for answering.
Yeah, I thought one of the previous slides were showing CD8 Tregs, but maybe I saw it wrong. If you can go back.
Actually, they were CD8 cells, just generally. So the total CD8 compartment. So we did have CD8 in there, and we could define the CD8 compartment, but as you know, they're a very small subset of the T regulatory cell compartment. So we really need to make sure we've got enough starting cells as we go down the tree of gating to make sure that we're absolutely certain of the results we're getting, which is what we're doing now.
Great, thank you.
Awesome, fantastic. I'll keep going. I guess mechanistically, we were very interested in these changes in terms of the Tregs and T effector ratios. So the next question that we had when performing this project was whether or not the increases in Tregs were actually functionally relevant, or whether that was just a numerical increase. So we wanted to see if, in the absence of Tregs, whether or not SRH therapy would still be efficacious. And the schematic on screen essentially shows the experiment that we conducted to test this. And we used a adoptive transfer model where we used NOD-SCID mice that don't normally develop type 1 diabetes because they're immune system deficient. And we transferred into these NOD-SCID recipients T cells that would cause type 1 diabetes. And following this transfer, we administered the treatment, which was either SRAGE or the control arm, the vehicle. And on top of that, we layered an isotype antibody, or an antibody type that would actually deplete the regulatory T cells, called anti-FR4 or anti-folate receptor 4 antibody. And essentially the hypothesis here was that we wanted to test whether or not, if anti-FR4 was administered, whether or not sRAGE would still work. And within the duration of the study that we conducted, seen in the very bottom left-hand corner, certainly we did see that when sRAGE was given without the Treg depleting antibody seen by the red line, none of the mice developed T1D within that study duration. However, as shown by the blue line, particularly the diamond data points, when mice were given sRAGE, as well as anti-folate receptor 4 antibody, which depletes the Tregs, about half of the mice actually developed type 1 diabetes, which demonstrates that in the absence of Tregs, sRAGE therapy didn't seem to be as efficacious. We might move on to the next slide. So we had a lot of preclinical data suggesting that sRAGE therapy may be effective and may be able to prevent T1D onset in vivo through a regulatory T cell mechanism. But we wanted to see if there would be any translational relevance, whether or not one day this type of data may have relevance to humans or relatives at risk of T1D. So we started experimenting with human T regulatory cells. And in this study shown on screen, we isolated some human Tregs. These were CD4 Tregs. And we co-cultured them with fluorescently labeled RAGE ligands. In particular, we used the RAGE ligand advanced glycation end products, or AGEs. And what we found on screen is that certainly as the co-culture progressed in time, the fluorescence intensity of the human Tregs increased, suggesting that they were either binding or uptaking the advanced glycation end products, the RAGE ligands. However, if the Tregs were co-cultured in the presence of a RAGE antibody seen by the blue data points, the fluorescence intensity certainly was nowhere near as high, suggesting that a lot of that RAGE binding of AGE was occurring through the RAGE receptor. And on the very right-hand side, we have a bit of a control line where we simply incubated the Tregs with fluorescently labeled mock protein, human serum albumin. And we didn't see a lot of fluorescence, suggesting that the RAGE ligand binding was occurring in a specific way, and it was occurring via potentially the RAGE receptor. We performed a few functional experiments as well in terms of human Tregs, and we did this just to confirm whether or not the administration of sRAGE would be able to promote not only the increased number, but actually the function, which is obviously also very important. And to do this, we performed a bit of a crude experiment. We performed a co-culture with human regulatory T cells, as well as conventional T cells or effector T cells. And within this co-culture, we administered AGEs, and we either gave the co-culture a vehicle administration, which was essentially just buffer, or sRAGE, which was sRAGE contained within that buffer. And what we did see, as shown in the black and the red bars, is that if the co-cultures were given sRAGE, seen in the reds, we had a significant increase in the proliferation of Tregs. However, if we look at the conventional T cell compartments, we see a corresponding decrease in the proliferation of conventional T cells, suggesting that certainly the Tregs do increase in growth, and they are able to suppress the growth of the T conventional cells as well. To layer on top of this study, we also used a few well-known inhibitors of typical T cell proliferation pathways, and these included the two inhibitors, wortmannin and tricirabine. And the rationale behind this was just to, one, use it as a positive control to confirm that these agents would decrease proliferation, but also to see if sRAGE would be modulating the proliferation of the Tregs through typical T cell pathways. And certainly we did see this, and at increasing doses of wortmannin and tricirabine, we do see decreasing proliferation of the Tregs, suggesting that the sRAGE effects do occur through standard pathways such as PAKT and mTOR. And the final study that we looked at least for this paper, was what the gene expression profile looked like in these human Tregs. And we certainly look at numerically an increase as well as functionally the Tregs are able to suppress T conventional cells. But why exactly is that occurring? What effects are RAGE ligands having on human Tregs at the subcellular level? Well, in this experiment, we administered a RAGE ligand, AGEs again, or the mock protein HSA, to human Tregs isolated in a monoculture. And following 72 hours of incubation, we collected the RNA and subjected it to a targeted analysis of RNA expression. And I think the technique we used was RNA nanostring. And what we see on screen is a comparison between AGE versus mock protein treated Tregs. And certainly there were many differences, as you can see in the top left-hand corner, via PCA. But if we really drill down, if we look at the volcano plot, we see there's a lot of downregulation of genes in particular. This is shown by the genes colored in blue. Some of these genes, the audience will probably recognize, such as IL-7 receptor, STAT5 and STAT4, KLRB1. FOXP3 is actually in there as well as GATA3, so as well as IL-4. So many typical T cell related genes, as well as genes which are well known to be critical for normal Treg function. But there really was quite a long list. So what we did was we performed an enrichment analysis, and this is seen in part C on screen. And we really did see downregulation of many well-known pathways, such as the IL-7 pathway, the IL-2 pathway, as well as the JAK-STAT pathway, which are very critical for T cell and Treg proliferation and growth. We performed an upregulation or upstream regulation analysis as well, just to see what impact these differentially expressed genes after AGE treatment would have on normal Treg function. And certainly many of these upstream regulators were critical genes for T cell function overall, such as CD28, IL-2, CD3, as well as IL-4. And just to finish off the bioinformatics analysis, we performed a network neighborhood analysis seen at the very bottom. And highlighted in green are the genes which are downregulated, and in red, the genes which are upregulated. And certainly many of the genes which were downregulated were actually direct adjacent nodes or neighbors to the FOXP3 master regulatory gene for Tregs, suggesting that in fact many of the modulated gene expression profiles have relevance to, I guess, the overall identity and function of human Tregs, which was good to see.
And so we might swap back to me now at this point. And so with respect to this research, we're actually looking at innovative ways to give soluble RAGE as a short targeted therapy or a therapeutic intervention to prevent type 1 diabetes. And so we're doing that in a couple of different ways. Can't really say too much about those two different ways, but needless to say that we are trying to find safe ways to give a short and targeted application of soluble RAGE. I mean, the great thing about the anti-CD3 therapeutic is that it's given as an IV infusion. And so in a nutshell, we could potentially piggyback soluble RAGE in there too. So that would be something that would be desirable, but obviously it needs to go through the proper regulatory process to ensure its safety profile.
So is that a clinical trial that's happening in Australia right now?
No, so the clinical trial that's happening in Australia is with respect to, we don't have any clinical trials running with RAGE at the moment. So I was going to show you something that we are hoping will move into clinical trials very soon for a few different reasons. And the soluble RAGE, we need to ascertain the method of delivery. So, which is what we're doing at the moment. So we hope to have that data pretty soon. This is a project that Kevin Herold, who you know was the first author on the New England Journal of Medicine paper. So he has also been interested in RAGE for a very long period of time. And so we had some very generous funding from the JDRF to look at RAGE antagonists. So these are orally available antagonists. They're available from a particular US company. And one of them had progressed to quite late stage clinical trials for a completely different indication. So it had already been given to some people with diabetes, again, a totally separate indication, and it could proven safe. And so this is a particular experiment in humanized mice. So these are mice with a human TB dendritic cell array. And when you give these RAGE antagonists on a model where you would normally see rejection of a transplant, so in this instance, this is a skin graft transplant, you can actually see that the RAGE antagonist definitely prolongs and stops that. And so that gives us a little bit of information in that these RAGE antagonists have the ability to modulate the immune system overall in humans. And certainly we've done some preliminary experiments with quite a low dose. We went a very low dose first, just to see, again, we're thinking about safety, but at the moment we're dialing these doses up. But you can see here that there was certainly some improvement in type 1 diabetes development with two different RAGE antagonists. So these are orally available tablets, which are easily taken by people to prevent the onset of type 1 diabetes. One of them, as I said, has proceeded to phase 3, so it's a much easier pathway. The other one is still in preclinical development and hasn't done its first in human clinical trials yet.
What is that, if I can ask, what is that a tablet typically prescribed for? outside of?
They're not prescribed. So the one in phase 3 failed. The indication that they went after, which was totally separate to what we're looking at, it failed from an efficacy perspective. So certainly passed all the safety endpoints and got to phase 3, and it failed for Alzheimer's disease.
Okay, great.
RAGE is a receptor which is thought to be increased in Alzheimer's disease. What's also important to note here is that we're trying to give quite a more concentrated dose at the start, and then to actually dial that dose down. So here, this is every day, and then we've dialed back to just a couple of times a week with the tablet. Again, trying to give the minimum dose to get the maximum effect. And again, we're looking at some other studies at the moment. And we also know when we gave these, when we looked pre-diabetes, so this is actually before the development of type 1 diabetes or autoimmune diabetes in this model, that we actually also saw improvements in first phase insulin secretion and blood glucose control, which was evident on glucose tolerance tests. And I'm happy to take questions about those things. We've got a lot more data, and some of it will be coming out for publication. So you will see some of that. We've been talking with the biotech company, and they're very generously agreed that one part of that can come out very soon. So we hope to share that with the community as soon as possible. And the second one obviously has some patent stuff. So we have to adhere to the wishes of the company in that instance. But I'm happy to answer questions if we can. But I hope today that we've shown you that RAGE is an important molecule that's present on cells of the immune system and in beta cells in type 1 diabetes, that agents targeting the age-RAGE axis could potentially delay progression to type 1 diabetes. We've certainly shown this in preclinical models, but we absolutely understand that many things which have been tested in there's some of these preclinical models have not moved into clinical trials and have not been efficacious in clinical trials. Soluble RAGE, we believe, elicits its protective effects via T regulatory cells, and they're probably the master at the end of the process, that soluble RAGE, and we're certainly investigating other parts of that pathway to understand what happens before the T regulatory cells go into the pancreas and do their tolerizing or whatever else they're doing there. And also we're interested in soluble RAGE and orally available small molecule RAGE antagonists as translating into the clinic for people with, or to prevent the onset of type 1 diabetes. Or even, we also believe that there may be some efficacy in new onset type 1 diabetes as well. So we'll probably be targeting both of those things as the regulatory pathway also demands as well. So thanks for your time. There's so many people who've been involved in these studies. These, I hope, are some of them. There are probably more, and I apologise if I've forgotten anybody here. But yeah, there have been some wonderful international collaborators involved in these studies, and I would like to say a very big thank you to them. I would also like to say a very big thank you to our funding organisations who have very generously provided the funding for all of these different types of studies, which have been going for a very long time. And certainly all of our participants and their families who have very generously provided samples for all of those original studies and for some studies that we're doing at the moment. So thank you very much for your time. We'd love to take questions.
Fantastic. We have a question here. What's the biodistribution of these antagonists?
So one of them has some published data. In terms of, so it stays in the circulation. So the pharmacokinetic and first-in-man studies are published for one of the antagonists. And so certainly you can find that. So that's aziliragon, so you can find that online. The other one has not been injected into humans, or, well, it's not been taken orally by humans. So I don't know what that looks like, but certainly the other ones are published.
Great. And another question, any undesirable effects, side effects expected from soluble RAGE or antagonists on other cells?
Yeah. So we know that RAGE is obviously involved in immune processes. So we need to be very mindful of that. And it's an important part of the pro-inflammatory processes. Certainly when you look at the late phase 3 clinical trials with oral RAGE antagonists, the only major side effect was gastrointestinal upset. So there was some stomach aches and things. There wasn't really much evidence for more infections and the types of things that you would expect if you were potentially modulating an absolutely key immune molecule. Now, we think this is an important molecule, but we don't think it's a master regulator in that if we really start dampening it, we're going to really, start to intervene in processes that we don't want to. The other advantage of this approach is that we also believe that there's a threshold where things like advanced glycation end products come in. We don't think they bind below a certain concentration, and that has to be met. And that we believe that if we target in the right way at the right dose, we can preserve the other pathways, which are important, like the HMGB1 RAGE pathway. Certainly, there is another clinical trial in Australia at the moment called BANDIT, and so it's targeting JNK1 and 2, so that JAK-STAT type pathway, and that's a major signalling pathway of the receptor for advanced glycation end products, among other receptors. And so it'll be interesting to see how that goes. So that's also a particular antagonist of that.
It's very interesting also, similar to teplizumab, you guys are advocating or going after a targeted timeline, right? It's a sort of a targeted attack or targeted therapy in a timely fashion, and then you're kind of like weaning away from it. So that's also very, that's very, from a user standpoint, that's very enticing. So you don't have to have it constantly throughout your life. Another question: have you ever tried to combine sRAGE and anti-CD3 in NOD mice? If so, any synergistic effects, efficacy-wise, or on the Treg phenotype?
Yeah, that's a really good question. So that was at front of mind. When we did the soluble RAGE studies originally, they were done a long time ago. And then we gave them to Matthias. You might have seen it was Matthias von Herath and the group at Novo Nordisk in the US who actually repeated the studies. We gave them the compound. We didn't tell them what it was. We just said, try these doses. And they came back and went, so what's this? So they were as excited as we were. And so we never got with this paper to that combination therapy, but that's an absolutely key question, because everything we do now in essence is probably going to be above and beyond anti-CD3 therapy. And so from the antagonist perspective, we're doing those experiments at the moment. So we're hoping to have that data pretty soon because I totally agree. That's a great question and really important one to address.
Great. Well, Matthias von Herath is heading over to head up the DRI in Miami, which I know they're very interested in, getting things to the clinic. So perhaps another collaboration is percolating. Who knows? And I'd like to thank you all again. Thank you both again, and everyone who attended. This is a fascinating new approach, and it has a lot of possibility. So we're wishing you all the best, and can't wait to be updated when the next set of papers comes out and that set of data is released. Really exciting times for you both.
And thanks everyone for their support, and we would love to hear your questions. Please email us if you've got other questions. We'd love to be able to go back and forth and get your advice as well. Obviously, we're only looking at one aspect, and we'd really appreciate any feedback that you've got. Thank you.
Fantastic.